on this 101 of the immune system. So we now get to another form of immune cell known as killer or cytotoxic T cells, a form of T cell. Killer or cytotoxic T cells. These release signalling molecules which kills the target. And then we also have T helper cells. So T cells are split into the killer or cytotoxic T cells and the T helper cells. And the T helper cells coordinate the immune response. Sensitivity and speed are features of innate immunity, whereas diversification and specificity are the key features of the adaptive response. To make effective memory, your immune response needs to be induced correctly. It needs to be primed during the initial stages of infection. In order for there to be activation of the innate immune system, will then result in T cell or cellular immunity and B cell humoral antibody immunity, which occurs in the blood. So it's like, if you think like T cell is the immunity that develops, is cellular based, whereas B cell is more sort of blood based in the form of antibodies. And this results in the development of a long-lived imprint of an event through a complex multicellular system where the info is not shared, where the information is not stored or retrieved at a single site. It's a whole team process. And there are, of course, many differences between us that might influence the quality of the immune response. This is known as genetic polymorphisms, or small differences, many sort of subtle differences, variability within a population, known as genetic polymorphisms, many different sort of forms, if you like, polymorphisms, different forms, many different forms of sort of genetic diversity. The most important area of the human genome is responsible for what is known as tissue type, or HLA type, and these molecules are responsible for presenting the peptide fragments derived from the pathogens to the T cells, and only those parts of a virus where the peptides can be bound into the groove of the molecules are visible to the cellular immune system, because the T cells are blind to the other parts. So this is the most diverse part of the genome. The individual's MHC molecules, as they're called, they vary in terms of the peptides they select for presentation, and this makes a big difference to the immune response. So certain MHC molecules focus the immune response on areas of the virus which are more effective targets, giving the T cells an advantage, and that results in a better outcome following infection for the host with that particular genetic sort of benefit, if you like. Now, it's one crucial aspect of the immune system referred to earlier is the need for the immune system to develop self-tolerance. Now, this is learned in the thymus, and this protects against autoimmunity. Now, immune tolerance, in order to develop immune tolerance, so the T cell will encounter self-antigens as it develops in the thymus. The T cells that fail this test are eliminated. What is required are receptors which are able to engage with the person's own molecules but not react against the self. It's a very important sort of balance, if you like. In fetus, the immune system presents a wide range of self-peptides in the thymus through the activity of specific genes. And this allows cells in the thymus to make proteins normally made only in very specialised cells in the body, for example, the brain, and then eliminate the T cells that react in this process of training. It's very clever. Only those in the narrow range of recognition to self, just enough but not too much, known as the Goldilocks principle, are able to exit the thymus. A lack of this gene is rare, but there are other causes of autoimmunity too. The system is necessarily leaky because it has to be flexible. The immune system needs to take a small risk in releasing T cells. The tolerance can be broken if a rogue T cell escapes the thymus, or if there's a very close mimicry between the pathogen and the self-protein. So what is recognised by the T cell receptor is a short peptide which is bound in the MHC molecule groove and presents only about 2 to 3 amino acids. Now, even if the peptides themselves differ between the virus and, say, a protein from the brain, by the time it's been presented in the MHC, the shape recognised may overlap. But the thymic system needs to allow T cells to escape, where such cross-reactivity may exist, or the ability to respond to pathogens is too limited. So it's a sort of like balance here, a sort of trade-off. So again, necessarily there will be some rogue T cells that escape. The tissues also protect themselves, for example, down-regulating T cells. There's an off switch, inhibitory molecules, and mutations of some of these genes can also result in autoimmunity. Now, tolerance. There's something called danger theory, which is important to mention here. Now, danger theory, the important issue is about whether the immune response is made against antigen. It's not whether the important issue about whether an immune response is made against an antigen is not whether it's self or non-self, but how it's presented to the immune system. So the antigens that are presented in a context of inflammation and infection will result in a response. But if there's no inflammation or infection, there won't be a response. The danger signals, the PAMPs, are sensed by the immune system. The absence of innate signalling resulting from the pathogen or inflammation means that the antigen should be ignored, whether it's a self or non-self. The suppression of function or the elimination of autoreactive T cells which have emerged from the thymus helps to, if you like, is one way in which autoimmunity can be prevented. There are some parts of the body which are necessarily tolerant. They have to be tolerant, and one of these parts of the body is the liver. Because we need to be tolerant to our own microbes living in the gut and the antigens from food, and all the blood supply from the gut drains through the liver. So presenting antigens against tolerizing environments is one way to limit responses, but it needs to be finely balanced, as if an invasive pathogen comes via this route, it is the ideal place to stop its spread. So again, it needs to be a sort of equilibrium. You don't want too much tolerance. But overall, it appears the system is quite heavily regulated and dampened in the liver. Hepatitis viruses exploit this tolerant environment in order to establish their viral persistence in the liver. A further environment where it's essential to damp down immune responses is in pregnancy, because the fetus and the placenta contain antigens from the father, and it's a mismatched graft, and it should be rejected. So tolerance mechanisms are in place, so the pregnant mother is moderately immune suppressed. Okay, so I'm going to stop there because there is quite a lot more I want to talk about. Well, I mean, I've covered most of it actually, and this is just a very rough, quick kind of run through the key aspects of the immune system, which are very interesting, and I will carry on this with you next time. So I hope you've learned some interesting facts today, and thank you very much for watching.